Hello everyone and welcome to a brand new video on DNA replication. So what we're going to be looking at in this video is a very mechanistic way of how DNA is copied and how it's copied during your cell cycle. So to do that, we want to first be able to look at a little bit of DNA structure, get an idea of what goes on in that structure of DNA and how that will allow it to replicate. And to get an understanding of that, we want to see the model that DNA is used, which is called the semi-conservative model of DNA replication. And we're going to see the places where it begins, the origins of replication. And we're going to see how we form what is called replication bubbles and how these bubbles form and how they end up releasing two separate strands of DNA. We're going to look at the enzymes involved, their role, and we're also going to look at a very important thing which is going to be related to that structure is the 3' and 5' ends of DNA and how DNA replicates in a certain direction. See how that happens, and then we're going to look at how we form two strands off of a template called the leading strand and the lagging strand, and we're going to see how that happens. So let's take a look at, first off, our DNA structure, and let's see how DNA itself is kind of built up. And to do that, we want to first understand the overall structure of DNA is made up of 1, 2, 3 individual piece parts that DNA is made up of. There will be a thing here called a pentose sugar. And when you see the word pentose, we're thinking 5 carbons here, a 5-carbon sugar that will be found in a DNA or an RNA molecule. We will also find a phosphate group, and phosphate is that PO4 compound called a PO4 minus, PO4 minus, 2 negative charges on that. And then we're going to see our nitrogenous base. And there is a nitrogenous base which will also be found at each what is called a nucleotide. So together these make up a nucleotide. Now nucleotides are the building blocks, the monomers of a large polymer referred to as a nucleic acid. Nucleic acids are DNA and RNA. Now, what is also to remind ourselves of is there is a pentose sugar and a phosphate that make the sugar phosphate backbone. So there is a backbone here, a sugar phosphate backbone, and the bases. So as we look at the structure of this, we will see that a phosphate group, PO4 minus, will bind to a carbon, and this carbon is going to bind to a ring structure. And this ring structure is forming our pentose sugar, and off of that we would see some nitrogenous base, a purine or a pyrimidine. We would have a base, a pentose sugar, and a phosphate group that make up our monomers of our nucleic acids. So remember, when we look at this, we saw there was a sugar phosphate backbone, so over here is our backbone. And the backbone is excluding our nitrogenous base. So let's look further at this pentose sugar. When we see the word pentose, remember we're saying 5-carbon sugar here. So we have a basic structure of a pentagram here, and we're going to draw that. But if you're drawing along with me, hold up, and what we're going to do is we're going to take our little eraser tool, and we're going to clip every corner off so that we can add some atoms here. And one of the atoms we're going to add is not a carbon atom, but an oxygen that will be found right here at the top. So you're already wondering, well, I see 1, 2, 3, 4 more places to put atoms on this ring. Well, those would be carbons, and you may be wondering, you said that I already told you that a pentose was a 5-carbon compound. So our last carbon, our 5th carbon, is going to go right here. Now, in these compounds, we're going to start numbering these. What you call these carbons, we refer to them as 1', 2', 3', 4', 5'. So the 1' carbon is here, 2', 3', 4', and the 5' carbon. And what you're going to notice is a very important thing. The 3' end of this pentose sugar, called deoxyribose, is sticking downwards here. And the 5' sugar here is sticking upwards. So what we have in our deoxyribose that is found in DNA is we have something very interesting here and very important that we notice. That DNA has a directionality to it where there is a 5' end sticking up and a 3' end sticking downwards. And this is incredibly important to understand, so we can understand replication. So this is what's going on here. So if we use this, we start at the oxygen and we start numbering in this direction. We run in that direction. We could say clockwise from the oxygen. Okay, so what we're going to start looking at next is the implications of this. If we take the sugar and use this, DNA is a double-stranded compound. Given that, a DNA molecule will be bound and have 2 strands, where one strand has a 5' end sticking out on one side and the other side has a 3' end sticking out here. Well, what's the implication of that is that the other side here will be opposite direction, where one goes in this direction and one goes in that direction. And we have a very important name to this. It is called anti, because they go in opposite directions, anti, but they also go side by side, so they're anti-parallel. Anti-parallel structure of DNA. Anti-parallel direction of DNA. So let's take this and put in right here where I have just lines, and let's draw some bases. So basically the lines that I drew that were parallel is representing our sugar phosphate backbone, and the lines going across it are our bases, base pairs that are binding through hydrogen bonds. So now let's just kind of come down and look at this, and let's start off with our 5' end, and let's build a DNA sequence looking at the bases. And I'm going to use one of my favorites because it is one of my favorite molecular biology jokes, is the DNA sequence for hairballing in cats. Cat gag. So cat gag, and it's always one of my favorite molecular biology jokes to make. So when these base pairs would form, they must form in a way that is said to be complementary. They're complementary. They must be complementary. Cytosine C is always complementary to guanine G. So our C to G. Adenine likes to pair with thymine T, thymine to adenine, guanine to cytosine, and all these have what we call complementary base pairs. So we have complementary base pairs, but because this strand is complementary, the 3' end must be there and the 5' end must be there. Remember, what is going on is we're numbering this based on the carbons clockwise from oxygen back up towards the carbon here. So we're starting here and moving here in that direction to number it. So we have two important things we just learned about. The antiparallel structure direction of DNA and how this creates a 5' end and a 3' end on one side, and then the opposite direction on the other strand. So we have two strands of DNA that we have. Now this is important because when DNA is going to be made a copy, we have two strands that can be used as a template. So if we come back to this and we start off with our 5' end here and a 3' end. Now I'm going to use black on all this, but because I need to be able to make a important point, our cat gag, that our 3' end is here, and of course we would have our complementary bases here and the 5' there. Now, what is extremely important for us to understand is that there is a model of DNA replication that we call semi-conservative. Now, there's a lot of information here. Semi means only partial, not fully. Conservative means I want to keep the old ways. So this means that when I make a copy of DNA, when I copy DNA strands to make two strands, that each new strand that I produce at the end of this will be half old DNA and half new DNA. Half old, half new. Semi-conservative. So what happens is I'm going to take this DNA that I produce, my cat gag and my GTA CTC, and I'm going to separate these two, separate them, break them in half, and use each of these base sequences here as a template to make some new sequences. So if I have my cat gag here with its 5' end here and 3' end here, let's clean that up a little bit, make that a little neater to read. Cat gag here, that will serve as a template, and then we're going to have our GTA CTC with its 3' end here and its 5' end there. These are going to be templates that are old DNA. This is the old DNA, and these old DNAs are going to be used, these old base pairs are going to be used as a template to come in and bind to make some new DNA off of it. And we're going to do this to make new DNA, and we're going to end up with two new DNAs who are identical in sequence to this DNA, but are going to be half new and half old. So if this is the 3' end here, the 5' end must be here, and the 3' end must be here. And we are going to be making complementary bases. So we're going to have cat gag here. And now we have made one strand identical to the original parental strand. Over here at the top, we're going to make GTA CTC. And we also have now made a strand identical to this one. So what we have done is made two DNA sequences who are identical to the original, but contain half old DNA and half new DNA, and are both anti-parallel, but both identical to the original strand. This is the semi-conservative model of DNA replication. So let's look a little bit more about how this happens, some more mechanism behind it. And I'm going to use the model here. I'm not going to discuss the differences between prokaryotes and eukaryotes here, but let's just look at eukaryotic DNA replication. Do know, you are aware they are different because eukaryotes have linear chromosomes, prokaryotes have circular chromosomes, and there are some differences in their origins of replication. But let's take a look at some DNA. And now what we've done is we're just going to draw a sugar phosphate backbone. So this line is our sugars and phosphates, and between here would be our bases, forming hydrogen bonds between both strands, the complementary base pairs of our DNA. Now, what is important is DNA, and as it comes to molecular biology, you need some signals that are going to let some enzymes know where to bind and what to do, when they need to do their job and where. So to do this, there are some signals that we're going to find here, and I'm going to put 3 of them here, and these signals are going to be telling some enzymes where to come in and start to pry the DNA apart. And these are called origins of replication. So let's just write ORI here, ORI sites, and I'm just going to write ORI here. It's easier than writing origin of replication. But these origins of replication in eukaryotic cells, the chromosomes in eukaryotic DNA, they have multiple. They can have thousands of origins of replication found on this DNA. So there's many of them. And what's going to happen is this is going to separate, and some enzymes can wedge in here and start to do something and separate it. So when this happens at these origin sites of replication, you will form a little bubble. And these bubbles are forming because enzymes that are involved, the enzymes of replication that we're going to talk about in just a moment, are wedging in there and separating these, forming what we refer to as a replication bubble. So a replication bubble has been formed, and what happens as we go in this bubble, new DNA is being formed here. New DNA is getting formed at these bubbles. And as the new DNA is getting formed, these bubbles, DNA is going to continue to replicate both directions from the bubbles. And as they do this, what is going to eventually happen is the bubbles will spread. And eventually, these bubbles will all touch each other. And when these bubbles all touch each other, these 2 DNA strands will separate. So these bubbles keep going until all the bubbles touch. And when they do, the DNA will now separate, and we will have 2 semi-conservatively replicated DNA strands who are anti-parallel to each other with their 5' and 3' ends. Just like we've shown before, but for simplicity, I want to show that the replication bubbles will continue moving both directions from the origin of replication until they all touch, creating 2 DNA strands that are identical. And if you're really kind of astute, you may realize that this is what will create the sister chromatids of a replicated chromosome, and why the 2 sister chromatids are identical but homologous chromosomes are not. So this is a very key feature to this process. So what we want to look at here is the enzymes involved, instead of writing all these out because it's just kind of hard to do sometimes on the iPad. So let's take a look at some of these enzymes that we need to know and understand to be able to do this. One that we're going to see is there's going to be, as I form DNA, there's going to be some fragments that I'm going to have. And we'll talk about those in a minute. And these fragments need to be joined together. They need to be linked And to link them, we use ligase. Ligase will link. Ligase links. To make new DNA, we need an enzyme to make it, and it's called DNA polymerase, and there's multiple DNA polymerases. I'm not going to go into the different polymerases here, like polymerase 3, for example, but they have the job of adding, growing the DNA. Then it has a second job as a proofreader. It's going to go through and say, oops, that's wrong, that's a mistake, that shouldn't be here, and hopefully aid in the base pair excision, base pair, improper base pairings to be fixed. Now, there's another enzyme called helicase, and helicase unwinds and untwists our DNA. Our DNA normally has a structure more like that, a twisted rope ladder, and helicase will help this unwind and unzip and open up the DNA to form what is called a fork, a replication fork. Now, as DNA is become released and such, it could fold over on itself and form self hydrogen bonds. We don't want that to happen. What do we want to do is ensure that when this DNA separates, that the only thing it binds to is its complementary bases on the new strand that we're going to produce. To do that, we add some enzymes to this, some proteins, stabilizing proteins, called single-stranded binding proteins. They prevent what is called hairpinning when the DNA strand binds to itself, creating a hairpin. Now there's, when this helicase here is causing an unwinding, this unwinding puts stress. It puts a stress on the DNA, a shearing force can be created. So ahead of the replication fork, we'll see an enzyme called Topo Isomerase. He prevents the overwinding. He's going to go in and break some bonds, break some phosphodiester linkages ahead of the fork and rejoin these so that they stay relaxed. Think of it like massage oil to help relax you, to help make sure things go smoothly. It's important to understand this because things like ciprofloxacin, for example, or Cipro, is an antibiotic use. One of the main things is we use to treat anthrax, and ciprofloxacin, and the other fluoro and chloroquinones are topoisomerase inhibitors. There is another one that's very important, and these aren't in any order, is an enzyme called primase who makes an RNA. It makes the primers of RNA, an RNA primer. And that's very important that we make these primers. So let's look a little bit about what these enzymes are doing and how they are functioning and related to this process. So we're going to start off a little bit of this. Before we actually get into the full mechanism of it, let's go back to our favorite DNA sequence, the DNA sequence for hairballing in cats, cat gag, and let's show a little bit of what's going on here with this and teach us some very important concepts that we have to know to be able to do this process. Now, we know that we have a 5' end and a 3' end on both of these strands, and that they are side by side, parallel, but they go in opposite directions, 5' to 3', 5', 3' that way, so they are parallel but opposite directions, anti-parallel. Why is this important? Well, first off, the enzymes that make DNA can only add nucleotides to the 3' end. We can only add to the 3' end. So, given that, we need a 3' end as a building block to build off of. So remember I said what happens to these is they separate, and so if I had my cat gag here as a template, with a 5' end and a 3' end, and I had my GTA CTC as a template with its 3' end and 5' end here, that these will act as templates to build off the new DNA. But we can only add to the 3' end of something. Now, here we have a problem. And for one strand, but not the other. Now, there will be a 3' end here, and there will be a 5' end here. Now, if this 3' end is here, and there is a DNA sticking out here, you can build that. And so what is going to happen here is you can add to the 3' end. DNA also only can grow in the 5' to 3' direction. So we can build this DNA in this direction, this way. So we build it in this way. So our C, T, C, and then remember here we have our T, our A, T, G, our GTA-CTC sequence here, and then on this one there will be a 5' and a 3' end here, and we're going to grow in that direction and make our sequence called catgag. And these are a very important set of things to know and understand, that so you can see that you have one strand growing that way and one strand going that way, which has a lot to do with why you have replication occurring in both directions away from the origin of replication, and why the bubbles form the way they do. So that's very important. So now what we're going to get into is really the most complicated part of this video, and how this all happens. How do we make the DNA? So to do that, and if you guys are drawing with me, it really will be very useful to draw with me on this. If you're somebody who's having a hard time understanding this concept, trying to learn it for your own courses, draw it with me here, and we're going to start off by drawing a replication bubble. And at this replication bubble, first thing we need to make sure we do is identify the 5' end and the 3' end here, the 3' end here, and the 5' end here. Now, it doesn't matter which way that you start. If you wanted to call this a 3' end, it's fine, but once you do that, you seal the fate of all the other ends. But we're going to do it this way for simplicity. Now what we want to do is add something very important here, is some hydrogen bonds here between some bases at these parts. The rest of them have been broken off. Now our next very big important thing, we're going to use some red lines here, and we're going to put our ORI sites, our origins of replication here. Now this is very important because this is where the enzymes had come in and started to form the bubble that we have here. So what we have is our replication bubble, okay, has been made. Now what we're going to do is start adding some enzymes. We said there is an enzyme that comes in that unwinds and unzips our DNA. This enzyme is going to be doing that. It is called Helicase. So I'm going to draw it as a triangle and put an H for Helicase. And it is moving in that direction. And just as it is there, there is another helicase over here moving in that direction. So the overall direction of replication is in both directions, away from the origins towards the forks is the overall directions. But let's see how this happens. Now, for simplicity as well, I'm going to add some little primers at some unique locations to help us understand it, but there will be some things I'll do that are a little bit different, trying to make it a little bit easier to understand than some of the models you may be used to seeing out there, but we'll put these primers at a couple places here. So one thing I want to do with some of my primers is we're going to put some primers on this DNA here. We're going to put some little what's called DNA primers. So let's add some primers, some little RNA primers, sorry. And let's add one right here, and this is an RNA primer. Now what is very important is they have some directions to them. There will be a 5' end sticking out here and a 3' end sticking out here, and you may be wondering why. The strand that we're going to be making here should be identical to this one. Okay, because we're making a copy. So let's put an RNA primer. Let's color him in a little bit here and write that in that this is an RNA primer. And if you're drawing along with me, make sure you put the ends here. Now, over here at this one, we're going to put one here, an RNA primer. And let's put one here. Now we have a problem here that this one here is going to be identical to this one. So there will be a 3' end here and a 5' end on that end of the RNA primer. And the other primer I'm going to put is also going to be here. And this primer will be, this one here will be identical, of course, to the one here as well. So this will be a 3' end, and that will be a 5' end. And the last primer I want to add is going to be over here. And this primer, remember, we're copying this strand here, remember, so it's going to be identical to that, so there will be a 5' end here and a 3' end there. Just like the first one we made, 5', 3' end. So let's add these 4 primers, and this is very important that we do this to help us understand what's going on. Now, also as a reminder, to keep these from folding over on themselves, there's going to be some proteins that come in and bind to our DNA strands, and these are called single-stranded binding proteins. Single-stranded binding proteins preventing hairpins, and we know they're there. Now, as this DNA is going to be made, remember our helicases are moving both directions from the origin of replication. As this is happening, this is creating shearing forces. To deal with those shearing forces, we have an enzyme, remember, that is before the fork, in front of the fork, and this enzyme was called Topo Isomerase. And I'm going to abbreviate him as Topo here, Topo Isomerase. And we're going to color him in Topo. We'll bring in as Topo Isomerase. And he's going to be correcting the overwinding. Now what we want to do is now begin to understand how we make our new DNA strand. The 2 rules were we can only add to the 3' end, and DNA replication will always be in the direction of 5 to 3. So let's start where we began. We began right here was our very first primer we laid down in my drawing. We have 5 to 3, as you're noticing, in this direction. DNA replication will always be in the direction of 5 to 3. Now, if we look at this DNA strand, it is growing towards the fork. Now, what we're going to see is, we're going to need some more primers in a moment, but this one is growing towards the fork. Now, it's going to constantly grow towards there. As helicase moves, it moves to cover up the area that helicase exposed, continuously towards the fork. This strand growing continuously towards the fork is called the leading strand that is replicated. Think about it this way. If you're in a race and think of the fork as the finish line. If you run continuously towards the finish line, you will be in the lead. The leading strand is the strand that goes continuously towards the fork. Now, when I go to form the strand above it, the other strand here, this strand, the one that we started building next, I have a 3' end here, and I can build DNA off of that, but I can only add a short piece of DNA. And what we have to do is have another primer here with a 5' and a 3' end. And then we will then use that 3' end to make another short fragment. And what you have is we are replicating. DNA replication will always be in the direction of 5 to 3, but we are adding to the 3' end to the 5' end between multiple primers. And we have made small fragments of DNA. These are called Okazaki fragments. Okazaki fragments. And the Okazaki fragments, kind of think of this like leapfrog. You lay a primer, build a fragment. You have a primer, do another fragment. In between each primer, we will build many fragments. So that means we're going to have another one of these somewhere else. And the one that uses the Okazaki fragments is being replicated by small fragments in pieces. So interruptedly or fragmentedly, it is built fragmentedly away from the fork. If you are running a race and you run and stop, and go in the wrong direction away from the finish line, the fork, you will not be in the lead. You will lag behind. This strand that we have made is called a lagging strand. Now, we've made half of it, and you can see that overall the direction of replication is going from the origin towards the fork because of helicase. Now, when helicase moves more of the fork, we will add another primer in that fork, and just keep working backwards. And that's how we would do that. So the other side, we've got the same kind of issues going on. I have a 3' end here, and DNA replication will always be in the direction of 5 to 3. We have our leading strand here. Here's another leading strand. And you will notice that, you can add to that 3' end, and we would have, if we have a primer on one end here, that if we try to do this continuously as a leading strand here like we did over here, I was trying to make a point here that we would have a problem that you can add to the 3' end, but we're going in a 3 to 5' direction, and we can't go that direction. So we have to erase that. And I was wanting to make a very important point here, and I hope I didn't confuse you if I said something a little wrong there. I wasn't wrong, I was just saying it in the wrong spot, I think, is that we'll need another primer. And we're going to add another primer here, and we add to the 3' end, yes, but we would be going in the wrong direction. So we will build these Okazaki fragments here and have a lagging strand. Is what I was trying to make is that when the leading strand is on this side of the fork, the lagging strand will be on the same side, the other side of the fork, on the other side of the bubble. So here we would have a leading strand going towards the fork on the other side. We would have leading strand. Now, I really hope this mechanistic view of it also helps you understand the overall process going on. I know this is quite a longer video than we're used to, but I hope my point that I was trying to make is one side is opposite to the other side was the point I was trying to make. So I hope you enjoyed this video and found it helpful and useful to understand the very complex topic of DNA replication. And the last thing I just want to mention is that if you're a very astute student, you're thinking, Mr. Lefford, these RNAs are in this leading and lagging strand. these primers, well, mature DNA doesn't have RNA nucleotides in it. So remember, we're going to have to come in with some ligases and remove these fragments, make sure that the primers are now replaced with DNA sequences, DNA nucleotides, making sure that there's no more uracils in that strand, and making sure there's no riboses, that we're going to replace our uracils with thymines, and we're going to come in and we're going to take the riboses and replace them with deoxyriboses using our ligases. Now, I didn't go through and put the polymerases and things in there because then it just makes a mess, but this concludes my video on DNA replication. I hope you found it useful, and I hope that you look forward to future videos from me. Thank you for watching. Please don't forget to comment, like, and subscribe, and as always, thanks for your support.